I'm here to talk to you a little bit about creativity in the sciences, or the subtitle is really how you tame curiosity. So I'm an assistant professor. I do bioinformatics, it's sort of like a blend of computing and molecular biology at the Copenhagen Center for Glycomics. And so we study sugars. So I guess I'll start off by talking a little bit about curiosity. And I guess it's a little bit funny that I've been asked to talk about curiosity because, you know, although I'm a little bit curious by nature, I don't think I've really considered myself a curious person. You know, I don't broadly find other subjects interesting. I'll give a proviso there, though. I do suffer from what is called a pathological curiosity. It's sort of described as a hypertrophy of curiosity. What that means really is that once I get curious about something, I can't stop being curious about it. I dive into it and I just go deeper and deeper and deeper, and it takes over. And I've become very, very specifically curious about one thing. And I think that's what I think about science. It's this unbounded curiosity about one particular subject with the freedom to dive as deep as you want into that subject to learn as much as possible about it. But I think there's a danger with doing such a thing. You could actually end up lost in your subject, not really knowing where you're looking, and not really knowing which direction to turn into. And on that theme of being lost, I guess we should ask a burning question, and that is, what exactly am I doing here? I was trained as a computer engineer in Australia, and somehow now I'm talking about my curiosity in my capacity as some kind of molecular biologist specializing in the role of sugars in a body at the top of a swanky tower in Copenhagen. And it would have helped a bit better to say that, you know, I'm missing the beaches when the snow is coming down. But it somehow feels that like my career path somehow went off track somehow. And, you know, I guess this all sort of went wrong when I accidentally started a new job, and it was in molecular biology, and I basically had no idea what I was doing. And so I got in and I started asking questions, and I got some answers, and then none of the answers made sense at all. So I asked even more questions, and I asked even more questions and even more questions, and then suddenly I became very curious about this area. And then that snowballed, and then this career in these sciences was born. Now, I've often been a bit of an outsider, and I guess this lack of baggage that I've had coming into the sciences and molecular biology from computing meant that I could ask all these questions from a very naive perspective. And it gave me a kind of intellectual freedom to ask all these questions and kind of gain some insights where maybe things haven't been thought of in that way before. And I guess it's the exploration of an area. So this is the deep field photograph from Hubble, which is basically overlaying a whole bunch of different images of the universe. This is basically what we can see in the universe now. And it's this free exploration of the universe, or specifically the unknown workings of the universe, that I guess is driving science. It's a driving force behind science. But what you won't see people doing in science, though, is sort of randomly walking around hoping to maybe stumble upon things. You need a guiding force. You need some way to be able to navigate this universe so that you actually be productive when doing your research, when you're actually being curious about things. And it's here, I guess, that creativity is vital in this space. What creativity allows you to do is to pick new and unique goals and targets to explore. You know, they don't actually have to be original ideas that you're searching out, but they have to be useful ideas. And then you can rely on the scientific method to actually allow you to validate that path that you take. You test it, you get data, and you test your ideas and your new theories against data and see if maybe you're heading on the right track. And this tether that you got with the data allows you to freely explore a space, and you can dive down whichever paths you think are interesting and know that you have something that can actually tie you back and bring you back out from that deep, dark place of curiosity. So we've gone through about 2 slides now, and I know it is early in the morning, and you are talking, I am a scientist, so I'm afraid I'm going to have to hit you with what exactly it is that I do in terms of research. And at the Center for Glycomics, we research sugars, or rather we study complex carbohydrates. Now, and I'm sure some of you are sort of expecting me to sort of come up here and talk about the simple sugars and how it's involved in diabetes and how it's sort of bad for you. But I guess I'm here to tell you that not all sugar is bad. And what you may not realize is that sugars are everywhere. And if you can find life, you can find sugars. They're a really fundamental biomolecule that is actually intimately intertwined with life itself. In fact, some people call the language of sugars the third language of life. So what exactly do I mean by that? Well, up here, you can see the first language of life. These are a bunch of chromosomes, and this is actually representing DNA, this first language. And it provides the blueprints. It tells the instructions to say, okay, what is it that we can make up in a body or a living organism? And you can think of it a bit like a big dictionary with a whole bunch of words in it and definitions for what you can do. Humans have around about 20,000 different genes. And then these genes then get translated into the second language of life, which is the protein language of life. So here you can see a protein here. This is the standard depiction of a protein structure. And the proteins go about and they do things in the body. They're the sort of functional units of the body. And you can sort of think of this as a bit like taking those words from the dictionary and putting them into a sentence, right? And this is where the sugars come in, because the sugars are the modifications on the proteins. They change the meaning or the function of the proteins in ways. And we can think of it like punctuation, or the way that you perform a sentence or read it out aloud. It's the difference for that sentence between "let's eat grandma" or "let's eat grandma." And it's this specific change in functionality that actually give the importance of the sugars. Because we think that the modifications in the protein are very important for determining their specific functions. And the sugars actually provide a lot of that capability to actually change roles of proteins. So here is one of our prettier figures that we use to describe what we call the glycome. This is actually a snapshot, a part of the entire glycome that you find in the human body and in a couple of different organisms. All the sugars are represented by these smaller colorful shapes. They're yellow squares, yellow circles, blue squares, triangles, diamonds, and all those kind of things. And it's the arrangement of these shapes into different larger structures or tree-like structures that actually sort of give us these complex carbohydrates. And there are about 30 different genes that actually control which of these sugars go onto different proteins. And it's where the sugars actually go onto proteins that really kind of modulate functionality. So after these sugars get put onto the proteins, they get shipped off into this thing called the Golgi Apparatus. And the Golgi is, I guess, a little bit like the factory floor in your cell. So it's this organelle sitting in the cell where these proteins come in, that they're immature, and then they get processed along this conveyor belt, and more sugars get added onto these proteins, and you get longer and more complex carbohydrates. And then that goes, and once all the sugars are finished and all the proteins are finished, they get shuffled off and sent into different parts of the body or different parts of the cell to do functions and to do things. And we know a lot about sugars, and they do a lot of different things. For example, and you may not know this, your blood types are actually determined by your sugars. So there's actually a complex carbohydrate that determines your particular blood type. And it's controlled by one gene, which is like this crazy gene, where you can have a whole bunch of small mutations in this gene, and it can produce a series of different structures. In cancer, you have one particular type of sugar that gets shortened, and you can use it as some kind of marker for cancer. Or when a virus comes along to your cell and tries to infect you, it's actually looking at a sugar first when it's trying to bind to your cell. And it's actually those little red diamonds in this figure here. So in total, there's a couple of hundred genes that control the entire glycome, and it has impacts over the entire body. So I'm hoping that maybe you're getting an impression of just the size of the space here that we have to explore. It's a gigantic space that we have to explore, and of course, we're curious about everything here. We want to know about how the whole thing works. And if we were left alone with our curiosity in this space, we would be lost. We would never come out and see the light of the day. And maybe my boss would be very happy about that because we'll still be in the lab, but we've got other things that we'd like to do. So how exactly do we tame our curiosity here? So I think that every scientist, in a way, lives for that moment of discovery. And I think that's sort of represented very well by this, the bathtub. And you think about that famous moment when Archimedes was sitting down in his bathtub and he saw the level of water rise, and he had these ideas about volume and density. And he jumps up and he shouts, "Eureka." And it's having that moment of discovery, realizing that some idea is right, is something that every scientist really kind of strives to do. And we recently had a moment like that. So we were trying to figure out what some genes were doing in the body. And we were characterizing them. And it turns out there was a pair of genes that actually didn't do what they said on the box. We looked at it and it was like, none of the results here make sense. So we thought, okay, we've got to find out what it is that's controlling putting these sugars onto this protein. The only problem was we had no idea really where to start. So if you remember, I said we have 20,000 different genes. So if it's not these two genes doing it, so which one of these other 20,000 is it that you actually use to find it? And the way we found it was essentially by looking at the equivalent of page 10 of the Google search results. So it was late at night, and I was very tired, and I've been searching around, and I'm going to be completely honest, I was already lost looking for another gene, and I was searching through some search results, and I was tired again, and the only thing that I could do, which I could think was productive, was to sit there just clicking on results. Click, 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 click. And we couldn't really, nothing really came up. But then one of the pages, something weird came up. And it didn't really make sense, but it was intriguing. So then you sort of look at it and you start reading a little bit more about it. But, it wasn't really kind of right. So you just sort of file it away at the back of your head and you don't think about it anymore. And then a couple of days later, it just sort of pops up again. And you start thinking a bit more about it. And in that time, your brain has been thinking about it and starts building models about how maybe this thing is right. And it's sort of a very tenuous connection there as to the idea that maybe this was right. And we think more and more about it, and then we discuss. And then we think, you know what, this is maybe the right thing to do. So then we use this technology called CRISPR. So CRISPR allows us to basically go in and edit the genome of a cell. So we go through and we remove this gene from the cell. And against all expectations, it's actually the gene or a family of genes that we actually wanted. So we actually were able to be creative in this space by relaxing our expectations about what the cell does, by relaxing our constraints and allowing our subconscious, in a way, to work on this idea, to be open and accept new thoughts and new ways of doing things, and then to work on it and focus on it. And then we actually went through and we tested it against data, and that actually confirmed the path that we took. So I think that was a good example of how we could actually use creativity to tame this curiosity space. So this process of using creativity to give direction in curiosity actually feels very important. And I'm actually very interested in this whole area. And I was very lucky to be involved in a project recently where we're looking at the intersection of art and science. And there's this great exhibition downstairs in the Vanderhallen, just at the sort of, anyway, it's downstairs. You'll find it. It's where we actually looked at, it's called Sugar Theatre. And it's basically a series of works that sort of explore sugar through an artistic perspective. And there are a couple of pieces of work that I'm really interested in down there. One of them is called Sugar Trees. And Sugar Trees I really like because it takes the front page of The Guardian and it sort of deconstructs the front page of The Guardian and takes the important keywords from the front page of The Guardian and then essentially puts that into the biosynthetic pathways that we're trying to establish from a cell and builds these trees with the words. And the idea is that you're able to read these trees and read the connections between the words and get an impression about what the world is like around you. So it's melding our ideas about biosynthetic pathways with some artistic representation. There's another thing called the Sugar Choir, where we've taken two sets of words of our jargon from the scientific worlds of basic metabolism research and glycomics, who don't really sort of sit together very often, and merge them together in this sort of harmonic choir presentation. And then finally, there's a poem called Sugar Contact, which I think really kind of exemplifies these ideas about this space between the world of science and the world of art. And I guess this was the first time that I was really interacting with artists, especially with Morten Sonnegaard. And I got a chance to really see their creative process. And it kind of made me realize that there's value in having open spaces to explore ideas. And that if you can figure out how to get over this communication thing, the fact that we're speaking different languages, the artistic language and the scientific language, collectively you can actually make something very, very interesting together. And for sure, the artists made some very, very interesting things. It was an experiment in creativity and how you can actually, the process of creativity is maybe common between art and science. So it turns out that this idea that there's commonality between art and science is not a new one. Carl Jung actually had something to say about it. And I ran across this when I was doing Google searches, again, curiosity and Google searches, for this phrase, pathological curiosity and hypertrophy of curiosity. And there was this really interesting essay, which I'd recommend reading, called Reflections on Art and Science by, and I may butcher this name, Aaron Katsier-Kachalski, who was a very famous biophysicist, and he died in 1972. And so he was quoting Carl Jung here, or paraphrasing him, and he said, "Both art and science derive their material from the collective unconscious, from the deep layers where the universal ideas of science and religion, of ethics and aesthetics, are living without contradiction and without competition." So, said simply, it suggests that maybe there's a common source for material when synthesizing new ideas in both art and science. It's that unconscious mind, that same place where you get ideas when you're having a shower. And you can imagine that maybe there are ways that by fostering creativity in both science and art and solving this communication issue, you could actually cross-pollinate each field with ideas. For me, I'm always eager to hear about new ideas because maybe it could give me new ideas about directions to explore. So here we are. Curiosity drives us as scientists, but it's creativity that actually gives us overall direction. We are, in many ways, reliant on serendipity to come up with the ideas that give us this direction, and we use our data to solidify the path that we take. But the question that we're not asking, and maybe the question that's being begged here is, can we actually do better than luck? Science is deeply curious because we're in search for the truth. And if we're not in search for the truth, then at least we're in search for the best model or the least terrible model that sort of describes natural phenomenon around us. and we want to fill in all the data. But then what happens if there's too much data? What if there's too much data for any one person to understand? What if the insights that maybe you can get from something is way beyond any single person? Are we doomed to be lost in the curiosity here? Are we just going to randomly walk around this space hoping that we get a new idea? Can we try and foster creativity? Do we know how to be really creative as a team? Are 2 minds better than 1? 3? 4? How do you actually get 4 people working in sync to be really creative? Or maybe, are we just treading water until we can have machines coming in and do the thinking for us? Is that the future of creativity? Can they handle this degree of complexity? Does anyone actually have any ideas about where we're going with this? I'd actually be curious to find out. Thank you.